Hi, my name is Jenalle Eck, and I'm a PhD student in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University. I'm at my graduate research site in Panama, a field station called Barro Colorado Island, a tropical forest in the middle of the Panama Canal. Panama's tropical forests are extremely biodiverse. The island alone is home to more than 300 species of trees, which is more tree species than can be found in all of North America combined. Ecologists like me have long wondered how so many plant species are able to coexist in tropical forests. Recent studies indicate that the microbes that cause disease in tropical trees, such as pathogenic fungi and bacteria, may play an important role in determining tree species diversity in tropical forests. One way in which pathogens influence tropical tree diversity is through a mechanism known as negative plant soil feedback. Negative plant soil feedback occurs when the pathogen communities that live in the soil of tropical forests change over time in response to the particular trees at a site. Pathogens are host-specific, meaning they are only able to infect certain tree species or genotypes, so pathogens proliferate near the particular tropical trees that they are able to infect. This pathogen buildup creates disease hotspots in the soil around large tropical trees, which then limit the growth and survival of the small seedlings that attempt to recruit into the forest. The seedlings that are most vulnerable are those that are similar to the feedback-producing adult trees, seedlings of the same species as the adult, or that share similar disease resistance genes. By limiting the recruitment of seedlings that are similar to the trees that already inhabit the forest, pathogen-mediated negative feedbacks promote tree diversity at a local scale. Despite the importance of negative feedbacks in influencing patterns of tropical tree diversity, it's unclear what factors determine feedback strength, and whether feedback strength varies among trees of the same species is not yet known. Tropical trees, even of the same species, vary from one another in several ways that may affect the buildup of pathogens beneath their canopies. Trees vary in the disease resistance genes that they possess, in their growth and reproduction, and in their biotic and abiotic environment. If trees of the same species produce different strengths of feedback for their own seedlings, then negative feedbacks may promote genetic diversity within tropical tree species, in addition to promoting species diversity within tropical tree communities. To test whether feedback strength varies among individuals of the same species, I'm conducting greenhouse experiments here in Panama. In this experiment, I chose 6 adult individuals of Virola serenamensis, or wild nutmeg, a common canopy tree species on the island. I collected seeds from all 6 individuals, germinated the seeds in sterile soil, and then put the seedlings into their treatments. I planted seedlings from each tree into pots containing the soil pathogens of its own parent tree, and also into pots containing the soil pathogens from each of the other 5 trees. By comparing the growth, survival, and disease symptoms of seedlings in each treatment, I'll be able to tell whether adult trees of the same species produce different feedback strengths for seedlings. In addition, this study could be among the first to provide evidence of genotype-specific negative feedbacks in a natural plant community. Insights provided by this research will increase fundamental understanding of the role of negative plant soil feedbacks in plant populations and communities, enhancing ecologists' ability to predict feedback dynamics and to include negative feedbacks in models of density dependence and tropical forest diversity.